Okay, yeah, I think I'll start now. Welcome everybody to the Seattle Quantum Computing meetup. Just a little bit of background. I created this group uh, yes, 2 years ago, I suppose. And the first year we met in person in the University District, and then um, this year we've been meeting online um, via Zoom. It's easier for people to access, of course, and so the whole purpose is to bring people together who have an interest in quantum computing. A lot of people, most of the people who have been attending the meetings are brand new pretty much to quantum computing, though not everyone. And so the idea is to just do some knowledge share and create some community and find each other. And last year, I became what's called a Qiskit advocate with IBM. And part of that program is to create community around you with folks interested in quantum computing. And so um, I introduced some quantum computing challenges last fall. And a couple of the attendees accepted those challenges and made some presentations in January and February. So I haven't yet decided yet on, that was great. That was really great. I haven't yet decided on the programming for April, but if anybody has any specific requests of things you'd like to see, you can let me know. For this one, I'm interested in quantum computing applications and algorithms. And so I started looking into the protein folding problem and how it's being addressed with quantum computers. And let's see, I have a question here in the chat. Were you a part of the Qiskit Summer School? I have been a participant in the summer school, so I was, I guess, a student in the summer school. So yes, in that way, I have. Were you? I'm reading the chat. If it others other than Milder are wondering. Yes. Okay. Awesome. So if anyone here has background already working with this, with the Qiskit code for the protein folding problem, you could very well have more experience with this than I do, but I'm doing this for my own learning and then also to share with others. So I put together this notebook based on some resources that I've been looking at, and I've just recently put it together. So this is the first time I've tried to present on it. I am recording it, and we do have a YouTube channel for the group, and I'll post this recording there afterwards. The January and February presentations are already out there. We also have a GitHub for the group, and I will put the Jupyter notebook that I created in the GitHub. Okay. So if you want to, I see everybody's muted, and that's the way I created the Zoom, is to have everyone muted on entrance. If you want to ask a question while I'm presenting the material, or if you want to add, just make a comment even, you can unmute and do that, and it will remind me a little bit that there's other sound out there. But it's totally up to you. I can go through the whole thing, and we can wait until the end also if you want to make comments there. Please go into, I'm reading the chat again. Please go into detail about QML, QDL, QMR, QRL, and AIF. Well, okay. I can't go into detail. I'm not going to go into details in all of those things because as the reason for that is I'm not familiar with all of those things. So yeah, I think, maybe uh, more about this, that maybe you could just take an opportunity to share with the rest of us what those things are about, or even like kind of where your question comes from. Yeah, I don't have enough background in the area to address those, so yeah, sorry. Okay, welcome, Sonia. Okay, so I'll get started here. So the protein folding problem, this is something that Qiskit is the IBM framework for writing code to interact with with the quantum computers and quantum simulators. And they have some code for this protein folding problem, which is given a sequence of amino acids which define the protein. Uh, how do you determine what its lowest energy structure, 3D structure is? And there are a large number of conformations that the protein, that these amino acids can fall into, and the protein itself in the body conforms very quickly. So it solves the problem very fast. Well, once the chain is generated, it folds very quickly. This particular protein that I selected here is called TDP43, and it has 414 amino acids, and I have seen some proteins, peptides selected in examples that relate to Alzheimer's, and this one potentially could relate to drug development for ALS, or motor neuron disease, and so I thought that would be an interesting area to pursue. So one thing is with Alzheimer's, aggregations are seen for a certain kind of protein, and there's a question of, does the disease cause those aggregations, or do those aggregations develop and then that causes the disease? And so that part of it is uncertain. So I think there's like a bunch of fuzziness, of course, in medical science about what's diagnosing diseases and then deciding how to treat them and how well do pharmaceuticals work. But I included here a link to a UChicago Big Brains podcast that was recently recorded, and there's a doctor scientist from University of Pittsburgh, Carl Harrop, who was interviewed, and the title of the podcast was "Where Has Alzheimer's Research Gone Wrong?" And he talks about this, about how it's been studied for years, and and there's still like there's not a pharmaceutical right now that treats it well or cures Alzheimer's, and so I think that's something to keep in mind when looking at these this protein folding problem. Just thinking about the protein folding problem and how it relates to actually translating into change in treatment. But so let's just continue going on here. And so the ALS, this TDP43, similar to Alzheimer's, it could have some relation to treating the disease, but I think there's still uncertainty there. Okay, so in this presentation, I based the material on several resources, and one is this new, I show a picture here, it's a new 4th edition of an IBM book called The Quantum Decade, and they have a section in there on the protein folding problem. This was part of the swag pack for the Qiskit Advocate program. Once you're selected, you receive this book and a very nice backpack that says Qiskit Advocate on it along with it. And The section there is pretty nice. It's has focus on various application areas, and one of them is this protein folding. And then there's a list of papers here that I'll bring up as I present the material. All these are links if you want to get the notebook later. Okay, so continue on. So in this book for the protein folding, they talk about designing a 3D structure of biologics and how that's important to function, specificity, and stability of the biologic. And so that would be what we're doing with the protein folding problem is predicting a 3D structure for the lowest energy state. And so there are different types of drugs, small molecules and biologics. And so the idea that IBM is presenting is predicting 3D structure of a biologic. And here we see it. I put some details about the differences between small molecules and biologics. Small molecules are largely what the over-the-counter drugs are, and the biologics are protein or other macromolecule like vaccines, blood and blood components, and so on. They're larger and they tend to be more specific in their action than the small molecules are. So we see here that maybe we expect the peptide chain to be greater than 40 or maybe greater than 50, depending on whose definition, amino acids in the chain for it to be considered a biologic. And so why would we want to try to use quantum computers for this? One reason is that classical methods like the AlphaFold 2 deep learning, there are certain situations in which those approaches don't do well. And then also the classical methods can't scale with the possible number of conformations in a protein as the chain gets larger. And we can see here a table of the number of amino acids in a chain and the number of possible conformations, and it's exponential in the length of the chain. So if we have 20 amino acids, we have a possibility of 109 conformations. 60 amino acids goes up to 10 to the 28, and 100 goes up to 10 to 47. And I got these numbers from the IBM Quantum Decade book. So, a biologic, if it would have to be over 40 or 50 amino acids, you can kind of see here that translates to a very large number of possible conformations. Okay, so we can look at a code example in Qiskit where we use their protein folding model. They have, based on this NPJ paper that I have listed, they have a set of classes that are now in the Qiskit research territory of the repo that you can import. And one of the largest class is the protein folding problem, and it's based on subclasses which are peptide and amino acid interaction. These are things you have to specify when defining your protein folding problem. And there are also penalty parameters that you can specify, which prevents the protein from folding back on itself when it's determining the conformation. And chirality is another property that's considered, and so on. So let's see. And then there's a method that the protein folding problem class has called qubit op, and that returns a Hamiltonian, which is decomposed into sum of Pauli operations, which can be used by the VQE algorithm. And so that's an overview, and we'll get to the code in a second. For the resources required, in terms of number of qubits, there are qubits that are used to specify the conformation, and they have described a sparse and a dense representation. In the dense representation, 2 times n minus 3 qubits are required, where n is the length of the peptide chain. And in this uh, basically toy example that I will go through, we're going to look at a peptide chain that has 6 amino acids in the chain. And you can see it there. And so that will require also 6 qubits. It's not 1-to-1, but it's using that formula 2 to the times n minus 3. And so this peptide chain would be classified as a small molecule rather than a biologic, but it's a starting point. Okay, and so that's for scaling. This approach has been identified as scaling O into the 4, and that is based on, I don't have this written out here, but this scaling is based on the number of terms in the Hamiltonian that captures the energy for the system. I'll put that, and later on I will show that in the paper they compared this method. They call it, this is the resource efficient method paper, and in the end I'll show a table where the scaling and qubit requirement is compared with this method versus other methods that were in the literature. This one was outperforming the other ones. So one of the simplifications that they make in determining the conformation of the folded protein is by using a lattice, a tetrahedral lattice, that the peptide chain can grow on like as it folds, and what's recorded is a turn, and therefore possible directions that it could turn. And then we also have some graphs here showing the resource requirements and the scaling for number of qubits versus the number of amino acids in the chain, and also the number of polystrings in the Hamiltonian versus the number of amino acids in the chain. Okay. Oh yeah, so here's the table, just quickly comparing the methods. This model is the far right column. And if we look at scaling, the next to last row, which is dedicated to scaling for each of the methods, we have n to the 8, and then we have exponential n to the 12, etc., and another exponential n, and n to 4. So we can see that this is the best, based on those numbers, this is the best scaling method. Okay. Also, this is a quantum machine learning example, and the algorithm being used is the VQE algorithm, but it is also a non-standard VQE algorithm, and they have named it as the CVAR VQE. VQE algorithm is a hybrid quantum classical algorithm. A parameterized circuit is run on the quantum computer, measurements are taken, and then an optimization is done on the classical computer to find a new set of angles to use in the quantum circuit. And for just a few details about the CVAR aspect of the VQE, it's what it does not, this method does not need an estimate of how it differs from the standard VQE is it does not need an estimate of the Hamiltonian's expectation value, but instead, it involves sampling of low energy tail of the energy distribution. And this difference between the standard and non-standard results in a drastic speed up to the optimization. Okay. So then, here we have the Qiskit code, and I can see here just, as this first cell is import statements, and you can see here it's coming from Qiskit research, as I mentioned. These things have moved. Qiskit, there was a Qiskit blog on Medium. I referenced that resource earlier in the collection, and they talk about how a little bit about the protein folding module and how it's moved and where you can find a tutorial, that kind of thing. The Qiskit blog has now moved off of Medium, but that article is still there. So yeah, one thing is you can see here in this import statement is this word interactions, and so this involves specifying there's different types of interactions that you can configure, and that's what the interaction is between the different amino acids. And let's see what else. As I mentioned before, there's a peptide class. Yeah. Okay, and just the protein folding problem itself. Okay, and then we have penalty parameters. Yeah, okay, so there's a fractal analytics paper that ran a bunch of examples using the VQE, different types of VQE, and also the QAOA algorithm that's listed in the resources. If you want to dig deeper into this topic, that would be a good paper to look at. I used their peptide chain that from their examples in this example, so I differed from the tutorial that's just offered in the Qiskit repo. And this is an Alzheimer's enzyme related peptide chain with 6 amino acids. And the next step, you can specify side chains, and I did not. And that simplifies it. These penalties, terms, you set them to values, and then use the penalty parameters class. The peptide, you define it by the main chain and the side chains. And then the protein folding problem itself, you define it using the peptide. You choose an interaction. In this case, I'm using the Miyazawa-Jernigan interaction. And then you set your penalty terms. And from this, you can get a qubit operator back out of the problem. And you can see it here. This is a sum of terms from the Hamiltonian for this problem. And you can see that it has coefficients times these capital letters. And these represent Pauli operators. You can see there's strings of 6 letters, and those are operators that will be applied to the 6 qubits that I mentioned before. Okay. And then, as I mentioned, the scaling depends on the number of these terms that we have in this sum. Okay, so this just shows the code that you can use to set up the VQE aspect of it, the sampling VQE, set up, yeah, you have to specify an ansatz. There's one built in called real amplitudes. I use that. Okay, and then you run that, ask it to compute a minimum eigenvalue, which is the ground state energy, the minimum ground state energy. And we see, I printed the result here, we get a result of negative 1.019, etc. And this is the same value as is reported in the fractal analytics paper that I mentioned that base their examples on the same peptide string. Okay, and so here we see a plot showing just the energy as we run the VQE algorithm. And we see it comes down and stabilizes to its minimum value. And then also from this, we can get the confirmation out and the way it, the folds that, the turns that were found at each amino acid. And you look at the plot here and I see a visualization of our final shape of this 6 amino acid peptide. And so it's just a very simple example. It's a tutorial getting you started example. And, but it's, there's so much involved in all of it that it's, I think, pretty exciting. And the question, is, well, for QML, for protein folding, what else can we possibly do? One thing I would like to try to do is pursue this in more depth and find, see if people working in the field of ALS could help me identify a peptide string that might be of interest for that disease area. And also, certainly, this is not really a real world example. So how do we move into use larger peptide strings, and how important are the side chains? What other algorithms could we try? As I mentioned, the fractal analytics ran examples with the standard VQE, the QAOA. Question is, can we define other non-standard VQE algorithms? Can we possibly use the iterative QPE algorithm? What other methods are in papers today that we could apply? And maybe just what if we focus on trying to come up with a new approach completely? And also the lattice model was used to simplify the process. What other there are other models we could try for that. Yeah, okay. Q-fold. I'm not sure what that is, Mild, but I see 3D rendering protein. Maybe you can explain more, share with us later what that is. Okay, so yeah, then maybe in the future, if people are interested, we could go more into the VQE algorithm. There's a very nice GitHub that Stefan Mangini created that shows the basics of what is this VQE. Here we call the sampling VQE available in Qiskit, but what if we wanted to write VQE from scratch? There's I listed a reference to the GitHub code for that. And so what we need for these problems is a Hamiltonian describing the energy of the system, and then we have to decompose that into a sum of these Pauli operator terms, which are the identity, the X gate, the Y gate, and the Z gate. And then when we decompose it into a sum, then we have to run a quantum circuit on each of those terms. And then we aggregate our results. And that's in the case where we are trying to estimate the mean value of the Hamiltonian. In this CVAR case, which is a non-standard VQE, as I mentioned before, we're looking at sampling of low energy tail of the energy distribution rather than trying to estimate that mean. So that would differ in that respect. And then there's a question of which protein folding problem should we attempt with quantum computers? And this is like, which problems at all should we attempt with quantum computers? And the answer is always um, we should use them for things that we can't approach with the classical computers. Yeah, and so that would be the idea here too. And there's an IBM quantum paper uh, came out in December 23 by Doga et al. And they addressed this question specifically, and they identify some protein folding issues that the AlphaFold 2 algorithm has had trouble with. I think it was certain kinds of mutations. They outline it in great detail. So if you want to learn more about that specifically, but they provide a framework for selecting your problem. So I think that would be if I can find a problem with this ALS, then I think that would be a good time to go to revisit their paper and dig a little deeper to see how it matches up in terms of if it might lead to quantum advantage to pursue that. And then finally, I'd like to come back to the beginning, and that is sort of this idea of, what is the problem? Are these aggregations of proteins causing the disease, or is the disease um, causing that to occur? And this idea that maybe we'd like to speed up research, speed up Alzheimer's research, that kind of thing. And so just the question of where do discoveries come from? And in the podcast, Carl Harrop, he says that discoveries come from left field. So I guess he's hoping for something to come from left field in with the Alzheimer's. And then also, I've heard David Oshalom at U Chicago in the quantum, working on quantum, talk about this kind of thing before. And I'm not providing an exact quote here, but but I've heard him say things about discoveries being accidents or kind of lucky occurrences. So I just like to keep that in mind, and then just like to think about, can we incorporate left fields and luck and accidents into our search, or to kind of maybe um, allow us to loosen up and maybe kind of um, maybe every once in a while allow ourselves to stand back, I guess, a little bit, and uh, maybe rethink what we're doing. Okay, so that brings me to my end. Let's see. I see there's a question here or a comment. Let me have to take a look. Miles 3 ring home. Mhm. I think, let's see, first of